Medical breakthroughs of the last century have transformed the human condition, but perhaps the most important one behind all of them was the discovery of the first immortal human cell line in 1951, HeLa cells. We've since made enough of them to wrap the Earth 3 times over. We know everything from the effects of radiation to drugs, running tests of the atomic bomb to cosmetics on them. Now where did these miracle cells fulfilling all these experimental needs come from? HeLa is the abbreviation of Henrietta Lacks, an African-American tobacco farmer. A mother of 5, she had advanced cervical cancer and came to Johns Hopkins Hospital for treatment. Without her consent, as was practice then, doctors took a sample of the cancerous tissue. But unlike all previous cells that would die within a few days of being taken from the body, hers continued to survive and replicate, presenting a panacea for research efforts. The impact was immediate. The following summer, a mass order was placed for HeLa cells to develop the polio vaccine, allowing them to end the worldwide epidemic. HeLa cells are the famed mother of virology, as scientists now learned about viral infections at the cellular level and created everything from vaccinations to treatments for tuberculosis, HIV, Ebola, cancer, and more. HeLa cells racked up an incomparable bragging list. They were the first cells to be cloned and to go to space. They were fused with mouse cells to create human-animal hybrid cells and were instrumental to developing cutting-edge microscopic imaging. What gives HeLa cells these special properties? Sure, they're cancerous and lack self-inhibiting controls on growth, but scientists never had these results with other cancerous cells. These are mechanisms we still don't fully understand, but have boiled down to 2 main ideas. Henrietta contracted HPV, which inserts its own DNA into host cells. DNA, which holds all our genetic information, is stored as chromosomes, of which every human cell holds 46. However, HeLa has around 78. The DNA insert turned off the p53 gene, a tumor suppressor, causing cancer and explaining HeLa's hardy proliferation. There is also the key discovery of telomeres in HeLa cells. Telomeres are bunches of junk DNA at the ends of our chromosomes, acting as protective caps for our essential genes. Through age and wear, they become shortened and the cells die. However, HeLa cells have the active telomerase enzyme, a molecule that continually lengthens the telomeres, allowing for indefinite survival, which may even hold the key for increasing our own life expectancy. Henrietta Lacks was never to know of these remarkable feats, dying 8 months after the sample was taken. The HeLa cell's genome was published in 2013, renewing research efforts, but also the controversy surrounding patient privacy and lack of informed consent concerning her immediate family. The legacy of Henrietta Lacks has shown how no science happens in isolation, and the conversation on scientific advancements and ethics will rely on our contributions to move forward. Thank you.